But actually, in the paper, at least I describe there's something wrong. I forgot about that. Homology-based inference for the conservation of enzymatic activity is the next topic. How well is enzymatic activity conserved? Can we use it for homology-based inference? And let's have a quick guess on this issue. So I talked about homology-based inference means that I infer for two proteins Q, a query Q, and an experimentally known protein, I have some level of sequence identity or similarity that is larger than some threshold. And if it is larger than some threshold, I say I can homology-based infer that when I know E, I can infer the localization or the feature for Q. That's homology-based inference. Now, there are two different features I put up here on the board. One is localization, and one is enzymatic activity. My question to you, if I sort of fix the level of precision, whatever that level is, you fix it, and my question then is, will that level then be higher? If I want to reach 90% precision, does the T have to be higher for localization or for enzymatic activity? So which of these features, so that two proteins have the same place where they are in the cell, or that they do enzymatically the same thing? What do you believe requires more sequence similarity? What do you lose earlier? Lose in terms of divergence, yes?
Functionality, so enzymatic activity.
Enzymatic activity, why?
Well, because it's localization, basically to encode localization requires less information, I would say, with signal peptide, for instance, because, you know, that's basically the postcode of
Good. Let's keep it there. We're going to go back to the single peptide. So it requires less information, but then you would argue on the note of what you say, less information. Single peptide is short, right? This is sort of where you're going with it. But the binding site is so short, right? An enzymatic activity site, in the most extreme way, you have a catalytic triad, which is 3 residues or something like that. So it's the most extreme version of an enzymatic activity site. Well, you need a little bit around it, but
also, if you argue like that, I would say localization could also be disrupted more easily, because it's, you know, if you say a signal peptide is short,
you knock out a few residues and you knock it out. In fact, that is a localization. We'll talk about that. We will talk about that. Yes, Paul?
Well, if we look at the two HSSP curves we looked at yesterday, one was localization and one was structure.
Yes.
And the structure one was better separated.
Yes.
And so you could say if structure implies function such as enzymatic function, then that should be easy.
That's an interesting argument. Okay. That's a very, very interesting argument. Now, let's return to what I said in the introduction. There are different levels of enzymatic activity. So enzymatic, the classification is the EC number. In terms of a tree, on the highest level, there are 6 different classes of enzymes: oxyreductases, transferases, hydrolases, lyases, isomerases, and ligases. And then we get into, that's the EC number. The first value will be 1. For the lyases, the first value will be a 4. 4.1 is the one that does a carbon-carbon, and so forth. It gets into further detail. Total number 4 levels. We do know for enzymes, there is this extreme example from an old paper from Janet Thornton, Dame Janet Thornton, where they show very nicely that you can sort of have a same active site, which is the one in blue here, and you see that underneath the structures are fairly different. Or that the placement of this active site in the molecules, the way this active site is realized in this structure and in this structure is very different. So you can actually, in that sense, back to what you suspected, Benjamin, and I'm not invalidating what you said in any way, but just this is an example that sometimes you can have a particular enzymatic activity realized by a relatively small thing that is put on 2 completely different, in this particular case, really 2 completely different proteins. Okay?
I wouldn't say that they're completely.
Yeah. I sort of see some similarity, but when you look at local similarity, you really cannot align them in 3D. So they chose this as an example to show, and when you sort of see how it is bumped into the major part of the structural core, if you want, in this particular case, it hangs somewhere completely different. So the active site, by no means, has a similar environment in terms of 3D structure in this particular case. And these cases exist. This is in some sense just one extreme case of that. In order to do this, we will do the same thing that we did for subcellular localization. We will take a database with all the proteins for which we know the EC numbers. And we will compare all enzymes of a known EC number against all enzymes with known EC number. And what we will get is something such as this one. So for identity class, so we're binning it, this is percentage sequence identity. It's not HVAL, it's not EVAL, it's just percentage sequence identity. And there's a paper from Alfonso Valencia and Damien Delvos. And blue is the structural thing that I showed you before. Essentially back to what Paul says. And now it gets slightly more complicated. So there are EC numbers here, and EC numbers 0 means that they're not even enzymes anymore. 1 means that the first class, lyases, is conserved, and so forth. So ultimately, wait a minute, the reading, this is structure. Oh no, this is the structure comparison, that's the enzymatic classification. And so essentially what this one says, until 85%, you have all 4 EC numbers conserved. Okay? Then you get to a level of sort of 55%, where at least the first 3 EC numbers here, you still have conserved. So, and you go on, and then you get down to a level of 25%, where you still actually can sort of classify. You begin to make a mistake here. The purple one is a mistake in the classification of this being an enzyme. But that's the only mistake you make. Now, I showed you a story. Yes?
I'm sorry, I did not understand it. The graph, could you tell me again what identity class is?
I'm sorry for that. Thank you for asking. Verena, the way now you go back to this story, right? You take all the enzymes for which you know the EC numbers, and then you compare the sequences of all these enzymes all against all comparison. Okay? That creates pairs that are such that for each A and B, so you create pairs Q and E, and for each pair Q and E, you know the EC number. Okay? Now, what I'm going to show you on the next graph is for percentage sequence identity between these Q and E. And they are binned. That means essentially that all the pairs with 95% sequence identity and more, they're in the first bin. All the ones between 85 and 95 are in the second bin. Sorry for that. Is that clear to everybody? And that one essentially is the percentage that have something in common. Here, the something they have in common in blue is essentially the same 3-dimension structure. Here in blue, it is essentially the entire 4 digits EC number. Yes? Ron?
So the SCC is, as well as ECC, some kind of structural determination of
Yes.
protein?
So this is sort of getting to the structural classification of proteins on the level of fold. So there's some classification.
That's not levels of its hierarchies and does also.
Yes. And I don't want to get into the detail because of this question. Okay. This is what we do. Is there any issue with that? Take all the enzymes of known enzymatic activity. We do the same for the structures. All the proteins of similar structures, and we compare them with all the proteins of similar structures. Yeah?
Since we talked about moonlighting proteins or enzymes.
That is a problem. Yes, that could be a problem, and in fact that is. But this is in fact. How would that problem, if that would be a major problem, what would that do to what I observe?
I can't really say because I still do not.
I'm sorry. So let's.
Because of the group, let's just look at the left graph. So
Yes. 85%. Yeah.
Yeah, it's a structural group of 6, let's say. I mean, it's a certain.
Let's just look at the blue. Let's look at the blue.
The question is different. The structural group 6 is not one group, but rather in the categorization you have a major group, a minor group, when you go back to the EC number, maybe it's
Okay, so there's a
clearer.
There's a hierarchy of protein structure where the lowest level here means that there is some secondary structure in common, or there is some distant similarity in the 3D structure, while the blue essentially is what you would call two proteins have a similar structure.
So, for instance, if a TIM barrel has like a 6-digit number, right, and then you have like two proteins that are both TIM barrels, and they have the same structure, they would be 95.
In principle, that is right, but let's just think about it in a different way. Forget the other colors on the left side. Blue means they have similar 3D structures. Okay? There's some classification scheme by which they have similar 3D structures. And that is called blue here. On the right, the blue means they have all 4 digits in the enzymatic classification identical. Yeah? Is that much clear?
Yeah.
Good. Now, where would your problem lead? If that would be a major problem, somehow, at least, so if that would be the only problem with what I see here, anybody can see where that would go in this graph? What would that, if the problem that many proteins are moonlighting or have different structures or something like that, yeah?
I mean, it would lower the scores of the...
It would lower the scores. So if that would be the dominant problem here, then this, you would never reach 100, right? Neither here nor there, or whatever you call moonlighting. So certainly not for enzymatic activity. If the dominant problem would be that too many proteins have two different functions at the same level of sequence, then you could not reach 100%. Right? So that is not the dominant problem. Yeah?
I'm not entirely sure, but one thing that I see here is that if we look at the structural...
The problem is not here.
But one thing there which I'm puzzled by is that if we have a 35% identity for the structural, but then for the enzymatic functions, and then there we have like the blue bars, they go until 35%, right?
Mm-hmm.
So that means the structures are the same.
Mm-hmm.
However, if we go to the function, we can see that well, at 35, at least a third of the proteins are at a different hierarchical level, hence at a different function.
Okay, so that essentially would mean, let's not do too much interpretation of this, but ultimately all we see is that the feature of having similar function you lose earlier in terms of sequence divergence. So first you lose the fact that so if two protein sequences diverge, first they lose function and then they lose structure. That's what essentially we see. And that is not a problem. That is in fact what we expect. Paul said what we expect is sort of a relatively sharp phase transition. Let's not get into that at this point. We will revisit that. The problem is somewhere else. The problem is here. Let's get to the problem.
Doesn't mean same function.
No, absolutely not.
All right.
Same structure does not mean a similar function. Structure is relevant for function. That does not mean that you cannot do a different function with the same structure. Structure tells us certain aspects of function, but then you could, for instance, have the same 3D shape and distribute the positively charged residue slightly differently. And you could no longer bind, or you could bind DNA. If you do it differently, you cannot bind DNA. For instance, this is one example, one extreme example, where you have two proteins that essentially have the same 3D shape. One binds DNA, one doesn't. Yeah?
So here we're comparing enzymes of known function against all enzymes.
Of all known function, yeah.
So of the all enzymes, we also know their EC number.
Yes, we do know all against all.
Yeah.
So we have a data set of enzymes for which we have the 4-digit EC number, and then we do an all against all alignment. We know the annotation. We just want to look up the sequence similarity so that we can bin them into these identity classes, right? What's the issue here? Bias, sequence bias. What we do is we essentially take everything and we're not asking at all. For PDB, what we know is that so in PDB at this moment, I briefly alluded to that the other day, when we look at PDB, that is about 200K. If I filter PDB at 100%, meaning that I kick out everything that is 100% sequence identical to each other, then I drop to ballpark 80K. So over 50% of the PDB, identical proteins, right? So establishing this, if I use the entire database, I completely overestimate how well these curves look simply because I look essentially at the same protein. Okay? Yes?
Wouldn't that only affect the last spin?
That would affect, that would not only affect, well, it depends a little bit on how you shape the families. But in principle it could affect every single bin, because the ones that are diverged pull in all the homologs of the other ones, right? Everyone that is distanced now, instead of pulling in one, pulls in many. So it will affect the entire spectrum. How is less clear, and I will get back to that issue. So that's the major issue. What we should do, at least that's what I try to convince you, or to hit you with in the localization story, was to begin with sequence unique subset and align against everything. And there I said, I'm going to explain to you why this makes a difference. And here is sort of one way of explaining it. And that goes back to Zipf. He's an American-born linguist and philologist who actually studied a while ago in the 20s in Bonn and Berlin to learn German, and found out, because he was polyglot, that in fact the word frequency follows this simple rule. And that this word frequency, that's sort of what the initial Zipf law is, that simple Zipf law, or the simple rule, holds in all the languages that he spoke, including German and English. So that ultimately, if you plot it, looks something like this. Okay? So there are some very frequent words and some words that are very unfrequent. Anyway, many words that are used less often, some words that are used more often. Or in terms of protein comparisons, you have some proteins that have a lot of binding partners and few proteins that have very few binding partners. Now, let's just look at something much simpler. Let's look at villages. So, if you were to plot the size of all the villages on this globe, so you have some villages you have many, many villages that are small, and you have some villages people call metropoles that are very large, right? How do you believe the distribution of that would look? It's a Zipf law. And this one is essentially the same Zipf law, except for we do here log-log plots, and then on log-log plots the same thing is sort of a straight line. And ultimately, if you do something in the computer, you just build your own artificial universe. All you need to do is to have something evolve in competition. You have a competition for resources. And then you always get the log-log. Sizes of bridges, or width of rivers, or whatever you plot. All of these things happen according to the Zipf law, in a straight line on the log-log plot. Yes?
Are those data points up there? They're like those outliers that artifacts up in the right corner. Yeah, this is just an error. It looks funny.
No, most likely these are data points, but why are they the outliers? So we're talking about they have a low rank.
Yeah. What does rank mean?
They are more frequent than they would be expected. They are most likely there's some mistake, because this is a pretty large city. Why would they? This is as if somebody says cities have to be that size, and we declare by law that there have to be several of those. This is the way it looks.
Yes.
It can only be an outlier. Yes.
Well, maybe these are ghost towns.
Yeah, but not that size. The log of the population is large. Oh God. Well, in Greece, well, let's not talk about it. Let's become too political on this issue. So I do not know the answer. Yes.
Sorry, just curiosity, because I've known this curve only as power law.
Yes, that is correct. It's actually exactly the same.
All right, fair enough.
It comes. It is a power law relation, and it comes from Zipf. I'm only going into the history of it. Now, my question to you is, what happens if you pick a random subset of a Zipf or a power law? What do you get?
You get a power law in the power law.
Yeah, exactly. You get a Zipf. So this is an interesting feature of that curve. Now, I want to plot the distribution of family sizes. So I'm going to have number of proteins per family versus how many of these families do I have. And I give you sort of, it could be flat. It could be sort of Gaussian. And it could be a linear line. What do you believe it will look like? Ron has an answer. Somebody else. Ron doesn't say it. So we have 2 answers, 3 answers, 4. Yes. Hello, why would I bring this, right? and again, so the subset. Okay, now we show you family size estimated by, in fact, doing what I call biased in red and unbiased. Biased, what I do is I take all against all. Unbiased, I begin with a representative subset and I align against all. Which one looks more like a Zipf, right? And this is ultimately in my eyes, this is the best proof to show you should never do a biased. So start with a non-biased set. Because something as trivial as family size, you cannot reproduce in any meaningful way. This is not a meaningful representation of the curve for family size. This is just one example how things would get absolutely wrong. And indeed, well, this is more art than science. So I assume that what I meant to do here is cut this one out. So ignore this. But this is still, there's still something wrong. This is why do I see it on the screen? Let's look here. Now things are a little bit more complicated, but in fact that is the right one. What I'm looking at is percentage sequence identity, percentage of protein pairs, and I'm asking, is the first digit conserved? Are the first 4 digits conserved? And this is essentially similar to the plot that I didn't understand initially, but it's just a redo on a slightly different data set, okay? And up here you have the biased version, and down here you have the non-biased version. And you see they're completely different. So the biased version would say, it is slightly different from what I had before, because this curve here before went further. But you still would say that at 20%, you still can infer all 4 digits of an EC number. While if you look at the unbiased version, you see you can absolutely not. The saturation point here essentially is random. So this is the point of random for this number of classes, 4 digits. So the random point here is different because you have only 6 classes. And you do see that there is a very sharp transition. And you do see that in terms of percentage sequence identity, this sort of seems to be further right than you expect from structure. Right? Everybody is with me on that. Now from here on it's too much detail, and I'm going to sort of it's slightly different for the different classes. What this one essentially says is you can do it for pairwise blast, for PSI-BLAST. PSI-BLAST does a little bit better. And it says that the best way to do that is use the H-val. And then you essentially get at this plot here where we compare the H-val, the log blast E value. That is the structure I showed you last time before, the very sharp transition. Subcellular localization is this blue. Now, the question that I asked, how would enzyme behave? So there are here 2 different answers. One in orange is the one when you want to infer essentially the first is you want to say it's a transferase. Okay? That's orange. When you want to really look at the activity, you look at the red. And ultimately the red and the blue are not that different. Except for so the random point here is a different one. Localization in this particular case was done with 3 classes. This is way more classes, so it goes down to something very small. And they are very unequally populated. That's why it does not go down to 1 over the number of classes. Okay, so this is why this is still 5% or 7% or something like that, because they're completely uneven. But anyway, what you do see is that some aspect of enzymatic activity, namely being a lyase, is conserved a little bit more than localization, and the real sort of binding site story slightly less. ultimately less because there's a sharper transition. And now, if you wanted to put a threshold to, say, homology-based inference, where would you put that?
I mean, if we're doing our main thing that we want to make sure that everything is correct, you have to put it at like, what, 40?
So something like this, yes.
So yeah, if you,
Now at this point, you would actually see that blue and red is similar.
then you can guarantee.
So the similar value for both of them, okay? Now, say, totally different story. That is what I would do, and again, you put it at 40, you put it at 60, you put it at 20, it's not that different. You have some wiggle space here, which is good. That means that your particular choice is not that sensitive to errors, right? Now, if you sort of push this threshold further, what's the incentive of pushing the threshold further down? So the price you pay is you begin to make more mistakes. But what do you win? Somebody else than Paul. Simply because Paul knows the answer. I want somebody to give me the answer who doesn't know the answer. Any suspicion? What do you gain from moving the threshold a little bit further? A little bit lower. So instead of picking 40, pick 20. Again, from 40 to 20, you make more mistakes. You make, I don't know exactly where this number is here. You maybe lie at 96, and here maybe this is sort of 95 or 94 or 92. So you lose a little bit in precision. What do you win? Now there are 3, yes?
Recall is fine.
Recall. So you have way. We have more points, and in fact, okay, I answer my my point already, and you see it already here. So the move from 40 to 20 brings up, in terms of percentage of some total here, tremendously, right? You more than double the kind of things you find. So the further you push, the more you gain in terms of this one. But of course, the more you also lose in terms of performance. But that is the incentive, to push this T as far to the left as you can. But again, the price you pay is that you pay in terms of precision, and there is such a rapid transition that you have to be a little bit careful in the way you do it. So unfortunately the message in some sense is, for localization and enzymatic activity, coarse-grained, the T is similar. When we look at more fine-grained issues, such as, is the protein part of cell cycle, and those kind of issues, unfortunately the story becomes much more complicated. And more detailed subcellular compartments than 3 states, again the story becomes more detailed. And I'm going to go through a set of slides. Whatever I do not discuss on the video is not going to be part of the exam, right? But now overall, the question is, how much can we infer through homology-based inference in terms of function for any organism? How would you guess that? How I gave you these thresholds T. How could you use that to estimate how much you can cover of a genome? So what fraction of human could you annotate through homology-based inference? How would you guess that? How could you argue from what I've shown you before? Let me put up, the problem is then I have to go over so many slides. How can I, from this slide, learn something about how many human proteins I can annotate? Not directly, very indirectly, and making a lot of guesses or approximations. But how could you use this T, or this establishment of something more than the green, more than structure, something that describes function like blue and orange or red here? How could you use that to essentially make a statement about how many human proteins could you annotate by homology-based inference? Or the sum of homology-based inference and experimental annotations. Any idea?